Okay, good morning, everyone. Can you hear me? I will try to keep your attention after the great presentation of Dr. Sean. I want to thank the Society for this invitation so we can show part of our resource at the Center for Discovery and Innovation in Northern New Jersey. And all of these resources are part of a paper we just published in Viruses Journal from NDPI. This study is based on our interest to characterize the new recent Omicron subvariants from SARS-CoV-2, which is the agent causing the recent pandemic, and also to characterize the infections produced by SARS-CoV when they are the single virus of SARS-CoV-2 or in combination with recent prevalent human respiratory viruses like SARS-CoV and influenza. So the virus was responsible for the pandemic, and the WHO ended the pandemic in 2023. But we have been seeing more SARS-CoV subvariants circulating until today. The real numbers from the WHO are scary. We have more than 778 million COVID cases and more than 7 million COVID deaths reported so far to the WHO. And here in this figure, we included the recent numbers reported by the CDC, which corresponds to COVID-19 illness, hospitalizations, and death that are still happening out there. In this diagram, we included the evolution of SARS-CoV-2 since the beginning in 2020 that you can see on the right, on the left. And then through the right, you can see the appearance of the most prevalent subvariants of SARS-CoV-2. And this group from Liu and colleagues in 2024 did a great job, including not only the subvariants that were prevalent, but also the number of mutations that were present in the different SARS-CoV-2 new subvariants. Most of them belongs to Omicron subvariant, which has been highly circulating in the human population. I also included this new diagram from the CDC, where you can see new subvariants that has been reported since that study in 2024. All the subvariants included in the blue circles has been reported and prevalent from 2024 to the current days. As you can see, there are many. And the other thing we learned from the SARS-CoV-2 evolution is that it's not always coming to the right, far from the ancestral strain. It's also going somehow to the middle, and you can see the MV.1.8.1 it was one of the most recent SARS-CoV-2 subvariants. Right now we have XFG accounting for nearly the 80% of cases that are currently infected with SARS-CoV-2. So SARS-CoV-2 is still ongoing, is evolving, and is showing new mutations, especially in the spike protein of the virus. So in our study, we selected some of the prevalent subvariants that were responsible for the ways of infection, and that's the graph on the left, where you can see a period from January 2024 to mid-2025. And from those subvariants, we selected some of them and did sequencing of the spike protein to identify the mutations that were present in those different subvariants. And as you can see here, we have represented by the dots with different colors, the mutations that were present in comparison with the parent Omicron strain, which is JN.1 in the second line. And what we observed is that some of the subvariants were showing new mutations not present in the parent JN.1, and also some variants like the last one, XDK.1, that were showing new mutations not identified in the previous relative subvariants. So we were interested in following up some of these subvariants, and we selected JN.1 because it was one of the most prevalent Omicron strains at the beginning of 2024. And we also selected KP3.1.1, which was also responsible for one of the highest waves of infection in 2024. So what we did was to perform neutralization assays. For this, we use 3 antibodies. One of them is just for research use, which is broadly neutralizing antibody, commercially available. And we did the first study where you can see the red arrow. In that study, we included Washington strain, which is one of the first strains isolated here in the US in 2020, and we used that strain as the ancestral strain because we couldn't use Wuhan strain. So in comparison with Washington, we evaluated different strains that evolved after that time until JN.1. And what we observed was that the neutralizing antibody failed starting in BA.5, which was like mid-2022. And it was a consistent failure until the JN.1 strain. So we were also interested to see what happened with the strains after JN.1, and if this lack of neutralization was also observed for the recent Omicron subvariants. And this is what you can observe in the top right graph and in the bottom graphs. So all the strains that came after JN.1, which belong to Omicron subvariants, they are showing immune evasion mechanisms, so the neutralizing antibodies are not effective. And this is what we have been observing also in clinic with patients that receiving sometimes antibody therapy. And the two antibodies we have on the bottom were used previously in the clinic, which are casirivimab and bebtelovimab. They are not used for emergency use right now, but they were used in clinic, and they are also failing against the new recent SARS-CoV Omicron subvariants. Besides looking at the mutations in the recent SARS-CoV Omicron strains, we were interested to evaluate not only the monoinfection with SARS-CoV, but also co-infection with other respiratory viruses in human. And this is because it has been underreported, the co-circulations of SARS-CoV-2 with other viruses like influenza and RSV, mainly because of the main focus was directed to SARS-CoV because of the pandemic. But we know, because there are some reports in the literature, we know that both viruses can co-circulate, and even you can have 3 viruses circulating at the same time in the same person. I will say the other reason why these co-infections are underreported is that because when a patient gets diagnosed with one of the viruses, it's weird that you get a second diagnosis for another virus. So you just receive treatment for the first virus that was identified in your body. So we selected influenza and RSV based on previous report and the high incidence of co-circulation with SARS-CoV. And basically, we were looking for a model where we can mimic what happened in the human lung epithelia to have a more reliable understanding of the mono and co-infections. And for this, we use human cells from the epithelia. These cells are provided by epithelials, where they have from the upper, middle, or lower respiratory tract. We choose the lower respiratory tract, specifically bronchial cells. And you can see on the left the composition of these epithelia. Basically, these cells can be grown in air-liquid interface model, where you can kind of replicate what happened in the human lung. So you have one part of the epithelia that is exposed to air, and the other part of the epithelia is exposed to liquid. So with these cells, we use transwell inserts, where you can develop this epithelia and try to mimic what happened in the human lung. The cells were cultured in the transwell inserts for about 5 days until we can see full confluency of the cells. Then we remove the media in the apical side, which is the upper side of the insert. And let me see if I can have, okay. So in the apical side, we remove the media around day 5, and then we leave the inserts for around a month until we get the full differentiation of the epithelia. Unfortunately, we couldn't include a video, but we can see the cilia movement of the cells under microscope, the clear movement of the cells. And then at that time, what we expect to have in the epithelia is this composition, where you can have all the cells that compose the human lung epithelia from the lower part of the respiratory tract, and it's basically formed by these cilia cells, cells that are responsible to produce the mucus, and other cells that make this epithelia functional. So what we did was to use different inserts with these differentiated epithelia and perform monoinfections and co-infections. For the monoinfections, we use the Washington strain, and I have included here the information of the viruses. We use the Washington strain, which we consider the ancestral strain, and we also monoinfect the epithelia with JN.1 and KP3, which were one of the most prevalent Omicron subvariants at the moment of this study. And we also use 2 other inserts to co-infect with SARS-CoV-2 KP3.1.1 and influenza, and the same SARS-CoV-2, but with RSV. We let the infection happen for 2 hours, and after 2 hours, we recovered the virus inoculum because we wanted to test the efficiency of the infection. And then what we did was to collect daily samples from the apical and basolateral side, performing watches with the same media. And those samples were processed by PCR specifically for virus quantification, so we can see how was the infection in the epithelia and compare mono and co-infections. And also by PCR, we determine the levels of the cytokine produced in the epithelia, because that's the other great thing with this model. We can have real expression of immune mediators like the cytokines. And we also perform immunofluorescence assays to evaluate the expression of the differentiation markers of the epithelia, where you can measure how much cells are responsible for the mucus production, for the cilia beating, and so on. So these are the initial results, so we can monitor the cytopathic effect in the human primary cells by looking at the epithelia. So on the mock cells, you have on the top, across all the time points, we performed infection and evaluation from day 0 to day 4 for most of the infections, but RSV co-infections were maintaining until day 6 because we know that's the time that requires the infection with RSV. We evaluated the virus inoculum we recovered after the 2 hours infection, and we think that the infection was successful because the amount of virus recovered was very low compared to the virus inoculum that we used at the beginning of the assay. And here, as you can see, I don't know how can I, okay, so on the second line, we have Washington strain, which is the ancestral strain, and you can see a little bit different compared to the mock cells, which were not infected, but it was not high infection compared to the infection with JN.1 and KP3, where you can see on day 3 and 4, we have a high impact on the epithelial morphology. And that was also confirmed when we used the co-infections of SARS-CoV-2 with influenza or RSV. And look at RSV at day 6, we almost didn't have remaining epithelia. Then we wanted to quantify the real infection, not just by looking at the cytopathic effect, but also the amount of viral particles in the epithelia. So what we did was from the apical and basolateral watches, we compared the amount of virus compared to the ancestral strain, but this time when we did the infection with JN.1 and KP3. And as you can see, we have a basal levels of detection of Washington strain, and I will say basal because you can see here at one of the late time points in the basolateral media, we didn't have more than 4 logs, but compared to JN.1 and KP3, we observed like a dose response for the infection with high significance on especially day 3 and day 4. So this was contradictory because when we were preparing this study and performing the assays, we were reading papers where people reported that Omicron subvariants infect better the upper respiratory tract than the lower. And we were surprised by these results. But those studies didn't include JN.1 or KP3, and that makes me think that probably the mutations we observed in these subvariants are responsible for this capability to infect the lower respiratory tract. And also when we co-infect KP3 and influenza and RSV, again, apical on the top and basolateral on the lower graphs, we also saw high infection extended until day 6 in the case of RSV, for example. This was confirmed by the expression of the differentiation markers in the epithelia. And that's the kind of confirmation we were looking for to make sure the infection was real. Again, here you can see the mock cells on the top, And then we have Washington compared to JN.1 and KP3, the 2 Omicron strains we use. And what you have here are different antibodies for the detection of the virus, or for the detection of the cilia cells, or for the detection of the cells that are responsible for the mucus production. And I can tell you, for example, that the antibody we used for the virus detection was nucleocapsid, a specific antibody for SARS-CoV-2. And as you can see, for example, in this column, in Washington samples, we didn't detect too much virus. However, in the JN.1 and KP3, we observed high detection of the SARS-CoV-2 virus. Also, we profiled the expression of cytokines in both the apical and basolateral side, and we observed a high upregulation of the pro-inflammatory cytokines, specifically in the samples infected with the Omicron subvariants compared to the ancestral strain, which also correlate the previous results where we were surprised, but they are true because we were confirming the results by different methods. So you can see here IL-6, interferon beta, TNF-alpha, and IL-10, which are most of the common pro-inflammatory cytokines after lung infection. And only for the interferon beta, we can see high levels in the insect infected with Washington ancestral strain. But most of the upregulation was in the Omicron, monoinfections, or in the co-infections. Similar result was obtained in the basolateral samples, where we observed mostly that the expression of these cytokines in the presence of the Omicron infection or co-infections with RSV. And this is just to show you the correlation in the expression of cytokines between the apical and basolateral side. There is a difference, a different expression in both compartments, which is expected because one of the part of the epithelium is exposed to air, the other one is exposed to the liquid with the media, and then cytokines are secreted and they are expressed also in the basolateral side. This is briefly because we only have 10 minutes, but we have a poster, number 18. Main remarks on observations from this study are that SARS-CoV-2 is still circulating. We are seeing new mutations in the recent Omicron subvariants, and I will say this will continue to evolve because the virus is showing immune escape mechanisms. And even when we have vaccines and treatments that they are effective, they need to be updated with the new mutations that the virus is showing in the spike protein. As mentioned before, we observed results contradictory with what is reported in the literature, but that means we need to keep the surveillance of SARS-CoV and continue to study subvariants, specifically those showing new mutations, because we need to understand what's happening with the virus. And the other thing is that the co-infection of SARS-CoV-2 with influenza or RSV, instead of attenuating SARS-CoV infection, we saw enhanced pathogenicity and synergy in the lung epithelia. That's our work. Thank you so much.
We have time for one question.
Just one more thing, and I forgot, and that's amazing. I need to thank my colleagues. We have here few team members, but all of them made possible this work. All of them are for the CDI, especially to Dr. Rashid, which is here with me. She works with me in my group, and she has done most of the work with Irene, especially for the ALI assay. And we have Kira, which is responsible for the PCR sample processing. We have Steve Park, and Dr. David Pelling, which is my mentor and also the director of the CDI. Now, thank you so much.